Good day, this is Professor Wills, ABMD PhD, and today is Tuesday, December 7th, 2021. It is 4:26 and 16 seconds p.m. Eastern Standard Time. Today's research, Darwinian principles. So let's hop to it here. Discovering new drugs with help from Darwinian principles. Date December 6, 2021. Source, University of Geneva. Our body must constantly defend itself against bacteria and viruses. It generates millions of different antibodies, which are selected to recognize the enemy and trigger the best possible immune response. The scientists use these antibodies for therapeutic purposes, to target proteins and disrupt their harmful. However, identifying the small molecules that will form the basis of the drug is a long and tedious process. The chemists have now developed a new technique inspired by the theory of Darwinian evolution, amplifying the best combinations and generating diversity allows biology to find solutions to new problems. The way drugs work is based on the molecular recognition of a target protein involved in the disease in order to disarm it. And to do this, scientists use high-throughput screening to identify which molecule could become a drug, specifically targeting the protein of interest. Over the last 10 years, the technique has been improved by encoding small molecules with DNA tags that simplify their identification, as DNA is easy to decode. And this is the principle of natural evolution, which consists of amplifying the best individuals while generating diversity to adapt and survive changing conditions. That is what researchers set up for small molecules. Indeed, the scientists have developed a technology that generates diversity by creating more than 100 million assemblies of molecules via their DNA, which they then select to best match a particular protein. Researchers were inspired by the characteristics of antibodies that recognize target proteins and sought to mimic them in the form of simpler molecules to allow them to be assembled in different combinations directed by DNA sequences. And these combinations are then selected and amplified several times to find the best possible match for the protein to be targeted, all in 1 to 2 weeks, compared with months or even a year for traditional high-throughput screening. And to validate the effectiveness of this methodology, the Geneva team focused on the PD-L1 protein, which protects cancer cells by diverting the immune system. And thanks to the methodology, they quickly identified an assembly that specifically targets PD-L1, confirming that it works effectively. And this technique, which is easy to reproduce in any laboratory in the world, costs only a few thousand francs, compared with millions for high-throughput screening. By exploiting the evolutionary forces highlighted by Darwin, researchers can now improve molecular assemblies and open up new space of possible combinations that has not yet been exploited in order to create new, more effective drugs. Good day, this is Professor Wills, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.